Hello, students, and welcome to my supplemental screencast on biotechnology. This is for your final exam. I forgot to put this in the final exam slides review. Sorry about that. Struggle was real. Okay, question 1. How do scientists make plasmids? And probably more importantly, what are plasmids used for? What are they good for? Stop the video, see what you can do. Okay, to get this, you had to remember restriction enzymes. These enzymes are proteins that do something in the body. Something's an ambiguous term, so what do they do here? They're going to cut DNA. These evolved in bacteria as they're cutting out viral DNA that was inserting itself into the bacterium. They cut at specific sequences. This one's cutting at GAATTC, and it's making sticky ends. It's cutting after the G, and then it's cutting down here, and it makes two sticky ends. So that way you can piece them together. So that's pretty cool. So you're able to piece back together the DNA. Here's a list, a little hard to see, but a couple famous restriction enzymes. So let's get back to it. You cut the DNA at specific sequences. You then cut out a gene of interest, say an insulin-producing gene or a gene for human growth hormone or something, and you can insert that into the plasmid. You use some binding agents. The plasmid is a small circle of DNA that's outside of the bacterial chromosome, but it's still expressed. The ribosomes still read it and make the proteins. So now you've cut out, let's just say it's the insulin gene from you, and you put it into this plasmid. You can grow it in bacteria, and then it can make insulin that we can use to help diabetics. So let's check out a couple of real-world examples. We've got some for blood clots in the heart, a protein that helps with that, human growth hormone, cleaning up oil spills, pesticide resistance, etc. So what was the big picture? We cut DNA with restriction enzymes. We're able to do the same thing for a gene of interest in a different organism, combine the two together into bacteria and produce proteins. Pretty cool. All right, so if you want to learn more about that, just take AP Bio. All right, next question. How do scientists compare DNA samples? Pause the video. Okay, we did this in class. It's pretty fun. You all did a great job with the micropipettes. It's one of the more fun labs of the year. So we're going to run some restriction enzymes through the DNA. Your DNA is different than mine. So if we run restriction enzymes through it, it should have different length cuts. So here's the blood stain. Well, you can look and see which one matches up with it. Well, this one is an identical match, number 3. So the suspect number 3 left this blood stain at the crime scene. All right, so if you need a little reminder, we just cut up the DNA with restriction enzymes. We loaded it into gels with the fun micropipetters. We applied current. All right, it's going to move towards the positive end because DNA is negatively charged. The smaller ones move faster through the gel. And so you got some beautiful pictures like this as well, and it's pretty cool. We run it against some known markers as well so we can know the distances that the restriction enzymes cut the bases. All right, so each of these will be unique for each of you all. That's also called DNA fingerprinting and gel electrophoresis. Okay, last little bit here. How do scientists quickly copy lots of DNA? This is the one we did the least in class, and we did not do a hands-on lab with this. So I would recommend you go to learngenetics.utah.edu, and you can do the online PCR simulator there. I put that in Google Classroom for you to check out. So here it is. We're going to heat up DNA. Remember that DNA is held together by hydrogen bonds, just like water. So if you heat up water, what happens to it? It eventually boils and breaks apart. Well, here we can heat up the DNA, and look, when it gets warm enough, the strands are going to split in half. Now we cool it, and we cool it to a temperature that allows the primers to bind. We're going to use DNA polymerase to add new nucleotides, but we're using a special version. It's from bacteria in hot springs that can withstand high temperatures. So then we can raise the temperature and let the Taq polymerase, which is DNA polymerase, extend those primers and run. If you do this about 25 to 30 times over about 2 to 4 hours, you get just from a single copy of DNA, you get billions of it. Right, so that'd be good, so that we can run experiments or test it, etc. So that was a PCR. It's kind of like a copy machine. Basically, you're heating it up, cooling it down, heating it up, cooling it down, heating it up, cooling it down. All right, students, best luck for exams. You're going to ace it. It's going to be great.